Now let's talk about maple syrup disease. Maple syrup urine disease is going to be an autosomal recessive disorder. So autosomal recessive, and it's going to be a disease of amino acid metabolism. Remember, that's our kind of ongoing theme right now. Amino acid metabolism. So here, we're going to see both copies of the enzyme alpha ketoacid dehydrogenase, which is going to be deficient. So our branched chain enzyme is going to be deficient. Alright, let's get this moved. This enzyme is going to catalyze the degradation of branched chain amino acids. What are going to be our three branched amino acids? Remember, these are going to include isoleucine, leucine, and valine. Now, you can remember this by the mnemonic, I love Vermont maple syrup. So we've got maple syrup here, beating our disease. I love Vermont, standing for our three branched amino acids. Typically, these three amino acids are going to be broken down into alpha ketoacids and then into acyl-CoA, which can in turn get broken down into acetyl-CoA and then fed into our TCA cycle. Now, this second step occurs with the branched chain alpha ketoacid dehydrogenase enzyme here. A deficiency in this enzyme will lead to an increased alpha ketoacid level in the blood, especially of the amino acid leucine. Also, a deficiency of thiamine, so vitamin B1, notice how I snuck in B1 as a cofactor here. This will also cause an increase in alpha ketoacids, since B1 is going to be used as a cofactor for this reaction. Now, why is branched chain amino acid disorder termed maple syrup urine disease? Well, affected infants are going to be normal at birth, but they'll eventually develop a characteristic maple syrup urine smell in their urine. So, for step one, if you hear the buzzword maple syrup urine odor, I want you to immediately think maple syrup urine disease, and then the branched amino acids that are going to be associated with it, isoleucine, leucine, and valine. There are also CNS defects such as lethargy, feeding difficulties, coma, and seizures. If left untreated, this disease can cause intellectual disability or even death in the first month. Now, the treatment for maple syrup urine disease is going to be a dietary restriction of these three amino acids and supplementation of thiamine as well as cofactor. You got a young boy that comes in with alpha ketoacid dehydrogenase deficiency and is unable to degrade which amino acids? Now this should be a slow ball for you, maybe even a T-ball for you. So, what are the three amino acids in maple syrup urine disease, or a disease that has an alpha ketoacid dehydrogenase deficiency? Remember, you got to think of your isoleucine, leucine, and valine, your I love Vermont maple syrups, which represent these amino acids. Alright, moving on. We have our glycogen regulation by insulin and glucagon/epinephrine. Now that we've finished going through the key disorders of amino acid metabolism, we're going to now transition into the regulation of carbohydrate metabolism. We're going to talk about insulin and glucagon, two important hormones that regulate the synthesis of glycogen in the fed and unfed state, respectively. Let's go over the mechanism of each. In the fasting state, the synthesis of both glucagon and epinephrine are driven by a low blood glucose. So both of these hormones cause the activation of adenylyl cyclase, which synthesizes cyclic AMP, which in turn activates protein kinase A. Specifically, do you remember what kinase enzymes do? Well, kinase enzymes are going to phosphorylate its substrate, and in this case, protein kinase A is going to phosphorylate the enzyme glycogen phosphorylase kinase, which activates it. Glycogen phosphorylase kinase, which again is a kinase, can then go on to phosphorylate the enzyme glycogen phosphorylase, which also activates it. So, The role of glycogen phosphorylase is to break down glycogen into glucose via a process called glycogenolysis, which is going to increase our blood glucose levels to address our low blood sugar levels. So our hypoglycemic state is going to be corrected by breaking down our glycogen stores into free glucose. Remember, glucagon and epinephrine cause the enzyme glycogen phosphorylase kinase to become phosphorylated, which is then going to go on and break down our glycogen reserves here. Now, on the other hand, what happens in the fed or hyperglycemic state? When blood sugar levels are high, insulin is going to be brought into the picture. It's going to be secreted from pancreatic beta cells. Insulin activates a receptor tyrosine kinase by causing it to dimerize, which sets off an intracellular signaling cascade, which results ultimately in the activation of 2 protein phosphatases. Remember what a phosphatase does? These are going to dephosphorylate both glycogen phosphorylase kinase and also glycogen phosphorylase, causing both to become inactive. Consequently, glycogenolysis does not occur, so glucose is going to remain stored inside the cells to prevent hyperglycemia. A less high-yield pathway occurs in the muscle, in which calcium here and calmodulin can activate phosphorylase kinase. This coordinates glycogenolysis with muscle activity, ensuring that enough glucose is going to be present to make ATP for muscle function. Now here's what it looks like all put together. We broke each of these down into separate pathways. Now sometimes it can get confusing if you dive right into this subject, so looking at this full picture here, make sure you break it down if you feel overwhelmed. We have 3 separate processes. We've got our calcium calmodulin process, we've got our glucagon epinephrine process in hypoglycemic states, low blood sugar states, and then in the fed state, we've got our insulin pathway. All of this is used to regulate glycogen breakdown. So this is kind of our takeaway message here. Glycogen, it's either going to be stored or it's going to be broken down into free glucose. Now, some general rules are that glucagon and epinephrine result in phosphorylation of enzymes, whereas insulin down here results in dephosphorylation of enzymes. This is just a general rule. These phosphorylated enzymes ultimately lead to glycogen getting broken down to produce some energy. So we just talked about how glycogenolysis is regulated, but let's go over what actually happens in both glycogen synthesis and glycogen breakdown, termed glycogenolysis. Now this is what glucose looks like. It has 6 carbons, which I've labeled 1, 2, 3, 4, 5, and 6. Now glycogen is a polymer of glucose, where its branches are connected by alpha 1,6 bonds, and linkages have alpha 1,4 bonds. So here is a glycogen chain that is being created. Notice how each of these are 6 carbon glucoses that are being linked together. Since we have numbered our carbons in our glucose molecule, the 1,6 and 1,4 indicate which carbons are going to be connected. This means that most of the glucose molecules are connected or linked between the 1,4 position, whereas the branches, which we'll see here, are going to be linked via 1,6. So I know it's a little small, but here's our 1 carbon and our 6 carbon, so this will be a branch off, while our links, our long chain links, are going to be between the 1 and the 4 carbons. 1 and the 4 carbons. So notice how our glycogen chain is going to be 1,4 linkages, while our branches, our offshoots, our side tracks are going to be 1,6 linkages. Now, as you can see, if you keep connecting with 1,4 linkages, you end up with a straight line, whereas the 1,6 linkages come off at an angle or a branch point. Now, why are we talking about all of these linkages? By storing glycogen in its linear form with multiple branch points, long polymers of glucose can be stored in a compact form. When glucose levels drop in the body, this stored glycogen molecule here, so this is going to be a glycogen molecule, is going to be reconverted back into our glucose, which can be used to feed the body and feed into the glycogenolysis, glycolysis, and then the TCA cycle ultimately for ATP production. What is the difference between skeletal muscle and the liver with regards to glycogen? In short, the muscles are greedy. So muscles are greedy and keep all of the broken down glycogen for themselves, whereas the liver is generous. It's going to make glucose for the entire body. Now, in skeletal muscle, glycogen undergoes glycogenolysis, so the breakdown of glycogen, to provide glucose, and therefore ATP, which will be used locally within that muscle during exercise. In the liver, on the other hand, glycogenolysis is not used to increase glucose locally, but rather to maintain blood glucose levels throughout the whole body, and therefore supply energy for not only itself, but the whole body as well. We'll now demonstrate the mechanism of glycogen synthesis. Where have we seen glucose 6-phosphate before? Hopefully you're thinking glycolysis, where glucose is converted into our glucose 6-phosphate. So first, our glucose 6-phosphate is converted into glucose 1-phosphate by the enzyme phosphoglucomutase, a long enzyme name here. Now, phosphoglucomutase is going to simply move this phosphate around the molecule. Remember, our mutase enzymes simply move a group around that molecule. So we move it from the 6-carbon to the 1-carbon. We're just simply shuffling that phosphate around. Next, glucose 1-phosphate can be converted to UDP glucose by the help of the enzyme UDP glucose pyrophosphorylase. Now, UDP glucose is going to be the substrate, or the building block, the Lego for glycogen synthase, which synthesizes glycogen by adding UDP glucose to the end of a building glycogen chain. Glycogen synthase is activated by glucose 6-phosphate. So if you have a lot of glucose 6-phosphate building blocks, you might as well store it in glycogen form. Here we see a lot of alpha 1,4 bonds that are being created to form a nice straight linear glycogen chain. The body doesn't want just one long, inefficient chain of linked glucose molecules, so what can the body do to increase storage efficiency? Once the chain has grown to about 10 glucoses long, so we'll pretend this is about 10 glucoses long here, etc., we're going to introduce branching enzyme. And this is going to create a branch point by forming a 1,6 bond with a previous glucose. So notice how up in the picture here, this isn't just one long straight molecule. We've got tons of branch points. So we need to use the enzyme branching enzyme. Now here we're going to take a glucose, add it to a 1,6 bond. We've been doing 1,4 bonds. Now we're going to do a 1,6 bond to create a branch point. So there are 3 main steps that are going to be involved in synthesizing glycogen. It's going to be, we need to create our glucose 6-phosphate, we need to create a building block, we need to transfer that building block onto our growing chain, and we need to add branch points. Now after a while, the body eventually creates a long glycogen chain, which can be stored for later use. Now let's deviate a little away from the first aid book. We'll still cover the material, but present in a slightly different way. Looking at this pathway, we see that glycogen synthesis steps are located on the right side. So we just talked about glycogen synthesis. Glucose, glucose 6-phosphate, or glucose 1-phosphate, our UDP glucose gets stored into our long chain. We throw in branching enzyme in there. So we just talked about that. So that's our right side of this reaction here. Now, let's take a look at this left side, which is involved in glycogen breakdown, or glycogenolysis. So we've got glycogen phosphorylase. This adds a phosphate group to a glucose molecule within the glycogen chain, and that's going to separate it from glycogen. Where have we seen this enzyme before? Oh yeah, that's right. That's the same glycogen phosphorylase that we talked about earlier, which is going to regulate glycogenolysis along with the enzyme glycogen phosphorylase kinase. So this can sequentially break down 1,4 linkages in the glycogen chain until they get to a length of about 4 glucoses left. This 4 glucose structure is called a limit dextrin. So a limit dextrin. It's going to be about 4 glucoses long. And then we've also got glycogen phosphorylase, which can't break this down any further. This enzyme's limited. To address this, we need to introduce debranching enzyme, which is going to be used to move all of the branch, except for 1 glucose, to the end of the chain, which allows for glycogen phosphorylase to continue and resume its function of chopping down this long built-up glycogen chain. So remember, a limit dextrin, we need to introduce our debranching enzyme to help clear the way, so that way we can continue breaking down our glycogen chain. In a smaller side reaction, notice how we have kind of an alternate pathway here where we take a look at the degradation of glycogen in lysosomes. This is going to require the enzyme lysosomal alpha-1,4-glucosidase, so here, which is known as acid maltase as well. As its name suggests, it can only break down 1,4 linkages, but it slowly converts glycogen down into free glucose via an alternate method. So we just take a separate path, a little detour, a longer detour, which is going to be a slower process because detours typically take longer than our normal glycogen phosphorylase pathway, and then also debranching enzyme. What are the end products of glycogenolysis? We just covered how glycogen gets broken down, but the end products of glycogenolysis in the liver is going to be free glucose and free phosphate. Now, if you remember from earlier, the muscles are greedy, and they're going to keep all of the glycogen breakdown products for themselves. Therefore, muscles break down glycogen into glucose-6-phosphate, which then enter glycolysis to make some ATP. So notice here, in the liver, we're going to create a free glucose molecule, which travels to the body. However, in the muscle, we create a glucose-6-phosphate, a glucose with the phosphate group attached. It's not free to leave the cell, and it's going to be then used for glycolysis within the muscle itself.